So welcome to the first virtual journal club. I'm super excited to do this with you guys. Journal club is kind of my favorite activity, and I hope you are interested in the topic. If you looked at the topic, boosters, the whole field, or this particular study, then you might have said to yourself, well, this is actually a pretty complicated issue. And the paper in particular is not easy to read. So unless you're a subject matter expert, it might take time to really dig yourself in. So based on these thoughts, here's our outline for today. We'll actually talk about the study goals first and then go into some length about the different combinations of primary vaccine series and boosters. And then we'll be relatively brief about the methods, and then I'll shed some light on the results and critically appraise them. And then I will take a brief glance also on what's been published since. And afterwards, we'll have lots of time for questions and comments from you guys for a hopefully very stimulating discussion. So what was known before the study? Also in the UK, there was another randomized controlled trial, the COV-BOOST study, where they tested the second dose in the primary series as a heterologous one. So if you got Pfizer, BioNTech, you got AstraZeneca, Oxford, or vice versa. And then there were also like 3 published full-length peer-reviewed papers and 2 preprints on the topic. And so what was known was that neutralizing circulating antibodies is significantly increased after homologous booster. But this was only there was only true for the Delta variant after an mRNA vaccine, after an mRNA booster. And then a preprint also confirmed that good neutralizing antibody response after a heterologous booster was seen. The T-cell response was good after AstraZeneca Oxford, but there were no data on any other ones. And then in terms of adverse events, they were similar to the initial series, except they were higher for COVID and CureVac. So here's some nuts and bolts on the COVID-BOOST trial, which appeared in the Lancet in late December, was published in an issue then and was online early December. It was a randomized controlled trial, multi-center from the UK. It had 18 sites. They were all in the UK. It was planned in early 2021 and then pre-registered in late April, and the UK is equivalent to clinicaltrials.gov ISRCTN. The entire trial, which with a planned enrollment of almost 2,900 participants, happened in the month of June 2021. The whole trial happened in June, in just 1 month. To be enrolled, you had to have your initial series, so the 2 AstraZeneca Oxford or the BioNTech Pfizer series at least 70 days prior to the dose, prior to the booster. And then there were several active and several control groups, and more on that in a little bit. It was double-blinded, so clinicians and researchers, as well as patients, were not aware of the study arm assignment. And let's talk next about the study goals. So this was a phase 2 study, which typically studies biological activity or a preliminary efficacy, which is then, of course, later tested in a phase 3 trial, and also safety. And so here they looked at immunogenicity, defined as the anti-spike IgG titer at 28 days. And they also looked at neutralizing antibodies, and it's the wild type, at pseudoviral neutralization assays and the T-cell response. And for the anti-spike IgG, they not just looked at absolute titers, but also looked at ratios compared to pre-booster levels. And then in terms of reactogenicity safety, they looked at adverse events, unsolicited, solicited, serious, and then also a moderate to serious. So which combinations of boosters and primary series were studied? The boosters studied here, and you'll have to bear with me and the study authors in terms of the abbreviations. So the first booster was the AstraZeneca Oxford vaccine, the chimpanzee adenovirus vector vaccine, abbreviated CHA. Then secondly, the Pfizer BioNTech vaccine, the Moderna NIH vaccine, but notably this was the 100 microgram dose. Currently we use 50 microgram doses for boosters. Then the other adenovected virus vaccine by Janssen or Johnson&Johnson. Then the vaccine that is not, where the manufacturer is not pursuing approval anymore, CureVac. Novavax, a new protein-based vaccine, and then Valneva, which is an inactivated virus. They only studied participants who had received Oxford AstraZeneca or a BioNTech after as the primary dose. And so in some other Western countries, of course, Moderna became more and more available over time. And in the US in particular, this was available from the very beginning in higher quantities. At 26 use was also initially offered as one of the primarily recommended vaccines in the US, but of course its use has declined since and is not recommended in the CDC anymore. And then, as I already mentioned, the booster dose for Moderna has since been reduced to 50 micrograms. AstraZeneca Oxford has not been approved in the US, and approval is not being pursued right now. And then, as I mentioned, CureVac is not being pursued for regulatory approval anymore. Novavax has only recently been approved. Valneva is still in the process. And then Sputnik V and the Chinese vaccines were also not studied. So this makes for a lot of combinations. So if you take the boosters here on the left with their abbreviations, they studied full and half doses for BioNTech, for Novavax and Valneva, but not for Moderna. J&J, Oxford AstraZeneca, and CureVac. And they also had active controls, which was Neisseria meningitis vaccine. And then they tested all these boosters in participants who either had received the Oxford AstraZeneca vaccine or the Pfizer BioNTech vaccine as a homologous series, so 2 vaccine each. So this makes for a lot of groups. This is 26 groups that they compared here. And this is how this looks in the paper. So you have a lot of boxes, and they actually assigned the patients to these different groups here, A, B, C, and D, and they had control arms in each. And I'll go into the methods a little bit more in a little bit. I already mentioned it was a double-blind, multi-center RCT randomization with permutated random blocks. Controls received meningococcal vaccine. Participants had to be 30 years or older, with either no, mild, or only moderate, well-controlled comorbidities, and a lot of them were actually healthcare workers. Clinicians, participants, and statisticians were all blinded. And then the vaccines were prepared out of sight, and they put masking tape on the entire syringe to conceal the dose, the volume, and the appearance of the vaccine. And then they drew blood. The primary endpoint, or the co-primary endpoint, was for immunogenicity at 28 days, but they also did initial blood draws. at days 84, 365, and then for subgroups at day 7 and 14. And then they did an ELISA for their IgG antibodies. They did a pseudoviral neutralization assay. They did T-cell assays. And then in terms of statistics, I'm not going to go much into this, but they calculated for a 90% power to compare a geometric mean concentration of the anti-spike IgG, and they used a Bonferroni correction. So in terms of the results, here's some baseline characteristics. As you see, patients were like middle-aged to older, and they differentiate in the results, in the other results, between under 70 and 70 and older. About half were male and female, and then quite a lot of healthcare workers, about a third or more, depending on each group. Most patients were white, and quite a lot of them had cardiovascular comorbidities, although this included hypertension, some respiratory disease, and a small number of patients with diabetes. So I'm going to skip to the antibody and T-cell concentrations. So this is the first result figure that they have for the cellular response, for the T-cell response. You see here on the left all the different boosters in different groups, and then you see the absolute T-cell concentrations, and you see geometric mean ratios in relation to the control, and you have everything stratified for under 70 and for 70 and older. And as you can see, for the T-cells, there is, compared at least to antibodies, not a lot of difference between the vaccines. Maybe you have slightly higher response for Novavax full dose, for BioNTech full dose, for At26, Janssen, BioNTech half dose, and Moderna. But at least compared to the antibodies, everything is pretty close together. This is all for the Oxford AstraZeneca primed participants. So they're all at AstraZeneca Oxford for their primary series. Compare this to the BioNTech Pfizer group. If you just compare these two, you see that they were similar to each other. Again, you have slightly higher responses for some, but keep in mind that these are relative responses to the control. And I'm going to go into the difference between that and the absolute levels for the antibodies in a bit. So here's the results for the antibodies, for the ELISA, for the binding antibody IgG. And you can see a little bit bigger differences here, at least in terms of the geometric mean ratios. So you can see that the Moderna vaccine and the BioNTech vaccine here had slightly higher responses. Here you see the geometric means, and this is for the Oxford AstraZeneca prime participants. And then for the BioNTech, you see lower geometric mean ratios for some groups. And I just try to compare that a little bit in terms of their absolute titers. So the absolute levels, absolute IgG levels, also differed for the different boosters. So this is the Oxford AstraZeneca primed group under 70. And you can see if you had AstraZeneca Oxford primary and then you got another booster, you were boosted from around 800 to something like 3,000 here. Whereas if you got Moderna, BioNTech, you had much higher antibody concentrations. So this is just one of the four groups, the Oxford AstraZeneca primed one under 70. And then just looking at the means without 95% confidence intervals, here you can compare all the different antibody levels. So first of all, in terms of the difference between over and under 70, you see that for some groups, for example, for BioNTech here, you have higher absolute antibody titers over 70, whereas for other boosters, for example, here the Moderna booster, you have a higher titer if you're under 70. Overall, you can see that the lowest antibody response after the booster were achieved for the two adenovectored viruses, Oxford AstraZeneca and Johnson&Johnson, if you had Oxford AstraZeneca as your primary series. If you had a BioNTech as your primary series, you had a much more robust response. And then secondly, you also had only somewhat mediocre responses for CureVac and for Valneva. So let's move on to adverse events. They use these radial graphs here to look at adverse events. These are severe and solicited adverse events, so pretty selective. And just to walk you through this graph, so you have all sorts of different side effects here in each wheel, and they're the same for all of these. And then you have the different colors here for the boosters. So green here, for example, for Oxford AstraZeneca. And then the left graph is always the Oxford AstraZeneca primed participants versus the BioNTech ones in the one on the right. And A, B, and C here just refer to the different groups. So on this graph, you actually don't see a response that is particularly high, but keep in mind that these are only severe and solicited events. So the only real booster response in terms of reactogenicity you can see here is Moderna with a lot of fatigue between 5 and 15%. After a primary Oxford AstraZeneca dose, and then the Johnson&Johnson dose here also between 5 and 15% for chills and fatigues. However, this is adverse events were more frequent under the age of 70, and so going a little bit deeper into adverse events. These are all moderate and severe solicited adverse events. And you can see here on the left, the Oxford AstraZeneca under 70 group and the BioNTech primed under 70 group, and here on the right, the 70 or older. And here you'll start to see some more frequent adverse events. So you have some response here to Novavax, still under 25%. Then you have like a lot of response here in terms of various adverse events for Oxford AstraZeneca under 70, but not if you're over 70 for both of these. For Johnson&Johnson, you have some adverse events that are close to or cross the 25% border. If you're Oxford AstraZeneca primed, but in particular, if you had a primary series of BioNTech, so somewhat similar to what we've seen in the previous slide with the Oxford AstraZeneca group. And again, we only see this under 70. And here on this side, you see quite a lot of side effects, both under 70 and also to some degree over 70 for CureVac and Moderna in various dimensions, and the highest here in line with other previous reported studies is pain and fatigue. And again, this is a little bit more if you were either under 70 for both primary series, or if you were 70 or older and you had the Oxford AstraZeneca vaccine as the primary series. So the authors summarize or conclude the results, conclude as follows. All vaccines except Valneva after BioNTech boosted antibodies and neutralized in their assays. And then there were substantial differences in the humoral and cellular responses. And then that, along with vaccine availability, will influence policy choices. What I would add is that antibody levels were boosted a little bit more by mRNA vaccines than adenovirus vectored ones, with Novavax somewhere in the middle. And then reactogenicity was higher for the Moderna and the CureVac, but not the BioNTech vaccines for any primary series. And then for the adeno-vectored boosters, Oxford and Johnson&Johnson, if you had been BioNTech primed. In terms of critical appraisal, this was overall a very well conducted double-blind RCTs. The method's description is brief but adequate. The only concern is that how far out of sight and by whom the vaccine doses were prepared before consumption. That might simply not have been mentioned in sufficient detail in the manuscript. And then since this was an RCT, it included less sicker patients. Only patients with mild to moderate, well-controlled comorbidities were allowed to participate, and there were a lot of healthcare workers, which might also be somewhat healthier. They only studied patients who had received BioNTech, Pfizer, or AstraZeneca, Oxford for their primary series, but of course, there were other available primary series in other parts of the world that were not trialed here. Importantly, for Moderna, the dose has been halved for the booster in terms of its reactogenicity, and then antibody and T-cell responses, importantly, have not been validated yet to any hard endpoints. So to infection or symptomatic infections, severe disease, hospital admission, intubation, mechanical ventilation, and death. We simply don't know if a boosting by a certain factor 2x, 10x confers more immunity in terms of these endpoints, or if you just need to reach a certain antibody titer. Since publication of this study, what has happened since? There was just a couple of days ago in the New England Journal a very interesting paper on homologous and heterologous COVID-19 boosters. This is the MID 2112 study out of Baylor, Texas. They did test all combinations of the 3 available vaccines in the US. So 3 by 3 equals 9. The Moderna dose here was also 100 micrograms, not 50. And as you can see here, the adverse events were color-coded or in gray scale, mild, moderate, and severe, relatively similar, except maybe slightly more severe events after you had the J&J booster here in the middle. If you had primarily received an mRNA-based vaccine, particularly the Moderna one, and then maybe some more headache and myalgia if you had the Moderna booster here on the left, if you had Moderna as your primary series compared to BioNTech as your primary series. In terms of the antibody response, the binding and circulating antibodies, they were quite a bit lower at baseline for the J&J vaccine, but also for the J&J boosted ones. There were also very few of the Pfizer patients and none of the Moderna patients who had no neutralizing antibodies at baseline. So when they received the dose, was there a difference between the Moderna and Pfizer boosters? I think that's very hard to tell, and keep in mind that we also use a lower dose of the Moderna dose now. They also reported a T-cell response, and I'm not showing them here, but they were in line with the antibody response. The mRNA vaccine generally boosted a little bit better, and in particular, there was a better response if you had J&J as your primary series. There was a trial of various boosters out of Brazil if you had the CureVac vaccine. And as you can see on the right, you had a very similar, these are like 4 bars for the 4 different boosters, very similar adverse event profile. And then you had slightly lower, in the top row here, the baseline, you had slightly lower antibodies with a CureVac as your booster compared to at26, Johnson&Johnson, BioNTech, and AstraZeneca. There, then, of course, we're beginning to see some real-world data. So this, for example, is a research letter reporting on a study done at the VA, the Veterans Association, and they found that severe disease overall was rare, and that heterologous boosters, in particular for J&J recipients, protected more for any infection, as you can see here by the adjusted rate ratios, and in particular for a severe disease, moderate and severe disease, compared to if you got a Johnson&Johnson booster. This is a new real-world study from the UK, and here you can see different times since you received either your dose 2 of your primary series or the BioNTech or the Pfizer booster. This is the vaccine effectiveness in terms of any kind of symptomatic disease for 2 different age brackets. And you can see that after the booster, you have a significantly higher vaccine effectiveness for both the BioNTech and the mRNA booster. If you only look at hospitalization for these age brackets, you can see that you still have vaccine effectiveness over 80% if you're 175 days out after your second dose of your primary series. And then you get a little bit higher response to the boosted. And this is the same only for death. So I'm going to stop here, and I hope that we'll have some questions that you might have. You could either put them in the chat or you could unmute yourself.
Ben, a question for you. First, thanks for a great presentation. That was really, really helpful. Earlier on in your presentation, you talked about the data behind antibody levels and immunogenicity with respect to its clinical correlate of death and hospitalization, things like that, and the link there being a little fuzzy. What, it looks like from the later data that you presented that they are correlating. What's your take?
I think that is the $1 million question. I think we primarily saw.
I'll expect a check in the mail then.
Absolutely, Jeff. We saw these phase 2 studies, and now we're starting to see some real-world data. Of course, the RCT studies have a population that's better selected, but they have a better follow-up, etc. So what I would be curious to see is either more real-world data with the Moderna 50 microgram dose, or as you say, correlation between association between antibody or T-cell response and these, harder clinical outcomes. I suspect that there will be one, but that again is pure speculation. I think something interesting is also that they looked at circulating antibodies, and you could also make a differentiation to the neutralizing, the IgE antibodies on the mucous membranes. And maybe if you get symptomatic disease, you have not enough of those neutralizing antibodies, but you still have enough IgG and T cells to protect you from severe disease and death. Maybe that's something where a booster it can help more than just the primary series, even if you're further out from the booster. So Shirley asked a question in the chat. What should we think about a second booster? I think that's an excellent question. I think second booster is something what we don't really have a lot of data from, but perfect segue. I was thinking that we should do a journal club on this. There is some interesting data out of Israel. There's some other emergent data, and I think it's very unclear if a second booster is necessary to prevent severe disease and death. You had other vaccines where you needed 3 vaccines. We thought that the primary series for them, hepatitis being one example, but maybe boosters, would protect you more from new variants, including those who might escape the immune system, and they might also protect more in general from symptomatic disease, although you're still protected from severe disease or death.
I'm curious, in any of these studies, do they account for patients who had developed clinical COVID throughout all of this process in terms of testing their immune responses?
Sorry, your question was if any of the participants had COVID?
Well, one would imagine that if you were enrolled in one of these trials and got your first shot of whatever you got, and then they measured your levels, and they got your second shot and measured your levels as a response, or your second and then your third in the case of the 2-shot series. If you also at some point in there had developed COVID, one would imagine that you sort of auto boosted again to some degree at least. And I'm wondering how the trials accounted for that.
So the COV-BOOST trial, the study that we just discussed in more detail, they did not look at that. Of course, this is a confounding factor, but of course, it's the power of the RCT that you have randomization and you have that effect equally in all groups, but to be determined.
More or less theoretical question about boostering in the combination of being once or twice vaccinated and then infected and cured or gone by the infection. We had the discussion recently in Germany. The status as being cured expires after 90 days at the moment by law, not by medical logic. Do you have any data about the immune response or the immunosystem fitness for people who perhaps take the most obvious case, who have been infected 3 times, never vaccinated? Is that comparable to being boosted? Do you know anything about data on that? Or does anybody of you know anything about data on that?
So
Thank you.
I can give a quick response, but I actually don't have enough data, or I don't have any data. So others feel free to chime in. I think that the immune response after an active infection is extremely variable. So it might very much differ with age. In higher age brackets, with a much shorter immunity, and then of course you have variants, and with new variants, whether they escape the immune system or not, you might also have a less conferred immunity from a prior infection. If I have relatives who visit from other countries, Thailand, Turkey, who are primarily vaccinated with AstraZeneca, come into the US, wondering if they should boost with Pfizer, Moderna, we should go ahead and say yes based on the data so far. What recommendations for the future boosting when they return home? So I think this is exactly what the trial looked at. You definitely had a more robust response after you were boosted with an mRNA vaccine, again with the caveat that they used a higher Moderna dose in the trial if you had a primary series of AstraZeneca Oxford. In terms of the second part of the question, future boosting, I think that's still up in the air. If we need a second booster, a 4th dose. I had planned to invite several ID and vaccine experts for this session. And for the next session, we will have somebody who is in the HIP committee. We were thinking of doing that session as an audio-only session on Twitter Spaces. If you're not a Twitter person, we'd like to invite you anyways. And just try it out. Maybe it's just easier to listen on your phone with the earpods in rather than to have to join a Zoom session. So please join anyways, and we'll send you details via email. But again, very much up in the air, and new data on the 4th dose, on the 2nd booster out, coming out every week. We'll end here promptly. Thank you all for attending. It was a real pleasure, and I hope to see you in the second one. And watch out for an email. I'm going to ask you which format you like better. Great to see you all, and have a great rest of the day or evening.